Very excited and honored to have our next presenter to discuss a topic that just always comes up and will continue to come up because of its importance and to really quite everything involved in research regarding GRIN2B-related neurodevelopmental disorder. I'm here to discuss gain and loss of function variants, as well as to discuss some other things regarding patient registries, is Dr. Steve Traynelis from Emory University.
Okay, it's really great to be here. I love coming to this meeting. We had one in Atlanta in 2018, and just wanted to comment that the GRIN2B Foundation is really doing a fantastic job. So that's the same view that everybody I talk to outside of my own lab has. So you should all feel good about the effort that you put in. Let's see, I have a couple of, if I can. Thought we were in business here. Whoops. Anyway, just a quick note, not that you need to read all these names, but it goes without saying. If I say I mean we. And if I say we, I mean they. And so these are the team of folks that I work with. And just a quick comment to Hongjae Yuan, a close collaborator, doing a lot of work, who's involved in everything we work on at Emory. Let's see if I can go forward. This is my Emory mandated competing interest slides, involved in a couple of startup companies. Have a grant from one pharmaceutical company, actually a second one that has just started, and on a number of scientific advisory boards. This is a slide I showed in 2018. I like this slide. I just feel it's important to mention that although we're off in a laboratory and you might think that we live a different existence, everybody I know involved in GRIN research is a parent first, and so this is not just not a comment from my perspective, but everybody behind the scenes who's working for solutions here. You know, they, if not walking in your shoes, they understand your passion and your desire for improvement in the lives of your family. Okay, we're going to follow Becky, who started us with Genetics 101. I did not know that's what she was going to call it, but I got a couple of slides here for Neurobiology 101. And I think many of you probably know this, so I apologize in advance if you've all fully educated yourself on this, but just to synchronize our thinking, we've just got a couple of slides here. This right here is just a Wikipedia image of an artist's view of a neuron. Here is an axon coming down, making a connection, and the central feature of the brain is the synapse. This is sort of a mind-boggling number. Synaptic density is 1 trillion per cubic millimeter, and that is far in excess of the best chip that can be cast in any die worldwide, and by multiple orders of magnitude. And so the brain has incredible complexity and ability to do computation. These synapses come in two flavors, excitatory and inhibitory. Excitatory tends to cause neuron 1 to tell neuron 2 that it should talk, and inhibitory quiets neuron 2. And so of those two types of synapses, we are just going to focus on the excitatory synapses. They use this neurotransmitter glutamate. It's kind of amazing. It's monosodium glutamate, just a food enhancer, but that is the main neurotransmitter in the brain. 99.99% of the excitatory synapses use that transmitter. It's released in this interesting fashion through these vesicles into this very narrow cleft, about 30 nanometers, diffuses across that, and encounters in blue these receptors on the other side. And the ones we're interested, I think you know, are called ion channels activated by glutamate. And when activated, they open a hole in the membrane and they trigger current flow. Positive cations, sodium and calcium, flow into the cell, and that will depolarize the cell and cause it to talk to its neighbors. And of course, these synapses are connected one to another to form circuits, very much like what you would find on a computational CPU chip. So these glutamate receptors come in a lot of different flavors. There's 18 genes. They encode delta amphocanate NMDA, named for various reasons historically. Obviously, we're going to talk about just NMDA today. And those NMDA receptors, I think you have heard, are encoded by 5 genes, GRIN1, 2A, B, C, and D, and of course today we're going to talk about 2B. Important in all sorts of normal functions, learning, memory, movement, brain development, basically anything you can think of, this NMDA receptor plays a role, and almost everything the brain does, NMDA is involved in. It's on virtually all neurons. I've got here just a stylized cartoon of that synapse. Here is that axon terminal. Here we've got gold vesicles filled with glutamate, dumping their contents into this narrow cleft. Those molecules transit that cleft in microseconds. They encounter these receptors and activate them. Here's a real-world picture of that. This is an electron micrograph. I've sort of grayed out everything so you can appreciate the axon terminal. There are the vesicles that are holding glutamate. All of this electron-dense material that the EM can't really resolve is actually the NMDA and AMP receptors bound to about 100 other single-gene proteins just on the other side. So just continuing, we're going to just jump into just a little bit of what we call electrophysiology. I've just got one slide here of this, and then we're going to revisit it at the end for gain and loss of function. So here's our cartoon, and here's our real-world picture. If we stick a metal electrode into a piece of tissue that we've isolated and are studying function with, we could shock that fiber, that axon fiber, and we could evoke release of all of these gold vesicles. When we do that, I've got a little gold cloud here. You see that glutamate rises in the cleft. If we could measure that and plot that as a function of time going this direction, you would have very low glutamate. And then for just a brief millisecond, glutamate would shoot up very, very high. It hits a millimolar for those of you who like to think about their old chemistry classes. And then within 1 or 2 milliseconds, it drops right back down. And this is the brain's effort to become a digital system, because digital computation is so much more powerful than analog computation. On the other side, if we stick an electron of some sort into this postsynaptic cell and we measure the current that flows, things are quiet until we shock that axon. And when we shock that axon, the glutamate is released. It binds to these 2 receptors. They both open. The AMPA receptors produce a current, cations flowing into the cell, exciting it, but it's also super brief, just a couple of milliseconds and it's all over. And NMDA, under certain circumstances, becomes activated, and these produce these long, slow, rambling currents that are carried by sodium and also calcium, an interesting ion that has all kinds of different roles inside the cell and can cause the cell to do different things. Okay, we'll come back to that figure, so sort of burn that into your head in a few slides. So these NMDA receptors, again, I think you have probably heard, they're tetrameric assemblies. They have 2 obligatory gluN1 subunits, or the gene product as Becky described from the GRIN1 gene. This is a protein, gluN1, and 2 gluN2 subunits. And because there's 4 different gluN2 subunits, 2A, B, C, and D, we can make 10 combinations of receptors. We can always have 2 gluN1s, and then we can have 2 gluN2s, 2As, 2 2Bs, a pair of 2Cs, a pair of 2Ds, or we can mix and match those 2s, 2AB, AC, AD, and so on. The receptors that will be impacted by an individual with a GRIN2B variant among those 10 are shown right here. And of course, it would be the ones that have 2 copies of 2B. Typically, you're very likely only to have 1 copy of a variant. But also these AB receptors, this is the main cortical receptor in most of your brain as you reach an adulthood. And then you have this other very interesting receptor, BD, which is also a very important receptor in inhibitory synapses, which I'm not going to talk about today. So all of those 3 would be impacted by a variant in some part of that 2B gene. So here's a little picture, and I guess a cartoon, an actual real structure of these receptors. And I just want to orient you, because I want to make a couple of points about how the receptor works, why variants will change a receptor, as Becky described, into working too good or working too poorly. And so if we start on the top, this is just the linear protein that's encoded by your gene. And so if we just took that long gene and we stretched it, start to finish, and we color code it, we have these interesting domains. And if we jump over here to the far right, these domains fold up into these rough shapes. That green thing makes a clamshell. The two blue parts make another clamshell. All these little yellow pieces make a bundle of these rods. We call them transmembrane helices. They actually are embedded in the cell's membrane. And then it has all these little red, flexible, rubber band-like linkers together. And then this is an actual crystallographic structure published in 2014, the very first description by Hiro Furukawa of an NMDA receptor, gluin-1, gluin-2B, and I've just color-coded one of the 4 subunits so you can kind of see how these cartoons map onto reality. And this is another cartoon, also published by Hiro, I think in 2020. And what he's tried to do here is show you how these clamshells are actually oriented in real life. And so you can't really see from this, all these ribbons in here from what the real protein looks like. But it gives a nice picture. There are 8 of these on the outside of the cell, and they're sort of in these side-to-side or back-to-back clamshell dimers. And so if we just look at that and think a little bit about how the protein works, we have this very nice cartoon, which is brand new, 2020. And so just as an aside, our understanding of how the glutamate receptor works has increased tenfold in the last 4 years. And so as the groups organized to understand GRIN variants, at the same time we're getting a lot more information. And so this is all, whoops, apologies for that. This is all protein. Here, and we got those 8 clamshells, that stylized cartoon. Here's glutamate in green. It binds into a clamshell, and the clamshell actually closes down around it. And you can see that when you do the right experiments. And after that happens, paradoxically, these little clamshells at the top open. And then something really surprising happens. You have this rotation, almost like children's gears that are interdigitated, where you turn one gear and all the gears on the plate next to it go the opposite direction. You have this rotation of the glutamate binding clamshell rotates up at the same time the top rolls down, and it creates tension on those red linkers, and that pulls open a hole, and that's what the current goes through. So there are hundreds of these machines at every Synapse, and they work on a microsecond to millisecond time scale. So you gotta think that all this is happening very, very fast and in an invisible atomic world. But basically, it's not an impossible machine to understand, and we just have these rotations, these forces, tension, and then opening of this pore. Now, Becky mentioned something about tolerance in genetic variation, and she also mentioned, by the way, it was fantastic to follow Becky because she laid the groundwork in a really nice way, and that was a super clear talk. She mentioned how we all are different in this room. We have variation that does not cause any sort of problems, neurological or otherwise. And if you were to go out into the public on the train station or at the airport, there are going to be people who have variants in GRIN2B all over that machine that I just showed you on the other side. This is a new analysis that it was first started in 2013, and we sort of modified it a couple of years ago. And what we've done here is we've mapped where variation in healthy individuals falls and where there is no variation in healthy individuals. And so this region in blue, nobody in the room here has a variant almost for sure in this region, but these other regions, we can all have variations, and so there can be a genetic misspelling, a missense variant, as we heard in the last talk, in any of these regions without causing any problems, but you can't touch this part of the receptor. And the GRIN2B gene is extremely intolerant across all the genes in the genome. It just doesn't have very many variants, and when they do, if they occur here, they almost always cause some sort of pathological consequence. And so the variants most likely to cause problems all fall in this region. And now if we just go backwards to our, whoops, I'm going to figure out how to go forward at some point. If we go back to our machine and we start thinking about the rotation of this and the pulling, that blue region is sort of the action zone. It's right where the tension is being pulled as these gears rotate against each other. So it's in a sense, no surprise that this is the part that you can't have a variation in without changing the function of this machine. Okay, so what do we know about the variants? As of November last year, there were 707 different kinds of variants we knew about in all of the GRIN families, about 260 for GRIN 2B. As we heard, most of them were missense, misspellings of that genetic code. We also have the frame shift you heard about and some others. They predominantly fall in 2A and 2B, with fewer in GRIN 1 and a handful in GRIN 2D. The number of patients can be estimated from a number of informatics approaches. This is one that was described in 2020, where there were numbers given of how many variants you would expect per 100,000 live births. This would be the number per year. And that number, depending on lifespan, will come up to somewhere around 5 to 20,000, probably north of 10,000 in just the US. And so this is a disorder that is on par in size with something like Dravet syndrome and a number of the other rare diseases that I think some of you have probably are aware of and looked into. Now, there are a whole bunch of very different neurological conditions associated with the GRIN variants, and this makes it a little bit challenging, and there is a way to understand that. We see, for example, in GRIN 2B, and many of the patients will have multiple of these conditions at one time, so the numbers are not just going to add up to 252. But most of the individuals will have some sort of developmental delay, and the missed milestones will turn into intellectual disability, and the children will fail to catch up. About a third of them show some sort of seizure disorder, and then we have some other indications. And so I think a really good question would be, why do variants in exactly the same gene produce such an enormously different spectrum of results? And this is my answer. There's probably many answers to that. But my answer, thinking about it from a physiologist's point of view or sort of a receptor biologist, is that when the variants occur in different parts of the receptor, each variant can change the way that machine works in a different way. And you can only really figure it out when you go and do a lot of detailed analysis. And if you go back to that cartoon where the agonist bound, the clamshell closed, the upper clamshell opened, and then it rotated, you could imagine a variant might change any one of those steps, and the result on the end function would be different. And so you end up with many different conditions based on where those variants fall. Okay, so before we go into gain and loss of function, any questions about that basic receptor function, kind of its structure? Yeah.
Specific to nonsense mutation or I guess deletion or null variants. My son he has a stop codon very early on in protein.
Yeah.
That's considered a loss of function, but that's a loss of DNA function to be able to produce the protein. But that just means that he has less receptors with the 2E subunit. So I think that's what's been confusing for some of us when we get a loss of function definition. It doesn't mean a loss of function in the good receptors that are there. It means that you have less of them in the brain.
Yeah, that's absolutely correct. And that's actually a good emphasis why this is a very unfortunate terminology, that it confuses people. But loss of, and as you'll see, loss of function is anything that decrements the overall function of that receptor in the circuit that I showed you on the first slide. And that could be a nonsense mutation that produces a stop, and the protein is not made, and it never reaches the surface, and so the cells just don't have as much. And that would turn up in the circuit as not as much. Another loss of function, as we'll see, could be something where the receptor just doesn't work as well, and again, they won't pass as much current, and it might, in a circuit level, be indistinguishable from what you mentioned, although very different mechanisms, but they still fall under this loss. So yes, a null variant is almost always a loss of function unless the stop is so far downstream in the protein nearly towards the end that the whole protein is made anyway. You just cut a little bit off and now it folds up and might actually change how the receptor works. Good question. Any other questions before I jump into gain or loss? Okay, so unfortunate terminology, gain or loss. Every protein in the body is really complicated, has many ways its function can change. That protein I showed you has 7,000 amino acids in it, so it's made of a lot of Lego bricks, if you will, and there's a lot of different ways its function could change if you put the wrong brick in the wrong place. A variant that changes some aspect of the overall function that makes a receptor more effective is called gain of function. A variant that makes some aspect of an overall function of the receptor less effective is loss of function. And the only real way to do this is to go off and measure the function. And we're lucky with an ion channel. It's not easy, but it's doable. You can measure current. We can make these receptors, put them in cells, stick in electrodes, and measure in great detail their function. Other individuals who have variants in different proteins that have more complicated roles, maybe a connecting protein whose whole job in life is to grab protein A, protein B, and bring them together. Well, there's no functional assay for that. And so those individuals have a really difficult time figuring out what the variants do. But for ion channels, we can measure current. And so we have a lot of functional data for them. Now, I have an analogy. I like this analogy. It might work for some of you. If it doesn't, I apologize for it. But there's many. If you just think about a car, there's a lot of ways you could assess the function of a car. We've got a couple of cars here, pretty different. You could quantify what a car does by how fast it goes, measure its velocity, its acceleration, its efficiency, its reliability. Does it brake? Ease of use, comfort, ability to carry cargo. There's a lot of parameters you could assess with that. And then if we took a car and we just think about a car as not a vehicle we use to go to the grocery store, but it's a machine. We just put on our engineering hat for a minute and we think, okay, this is a machine. It's got a lot of parts. It's got more than 6,000 parts. Car's more complicated than an NMDA receptor. And if we just pulled out the engine, and then we blew the engine up into its parts, and then let's just imagine for a moment on the assembly line, the worker reaches in the bin for a distributor, and for whatever reason, the wrong distributor is in the bin, but it just happens to fit on the car. He bolts that distributor onto the car, and he has installed one wrong part into that vehicle. And it goes down the line, and it gets finished. That part could do a couple of things. You could go take that car and maybe just by luck, putting in that particular wrong part, if it enhanced in any way how the car worked, we'd call it gain of function. Maybe it would make the car faster. Maybe the car would become more fuel efficient. Maybe the car battery would never go bad. Maybe it wouldn't break down. It wouldn't matter which of all those different modalities it changed. If any of them got better, we would lump it into this catch-all term, gain of function. By contrast, if you just jammed on the wrong part and you made that part swap, in any aspect of the car worked worse. Battery ran out every 2 months, had a hard time starting the car in the rain, take your pick. We would call that loss of function, and again, any decrement in any of the modalities of what that car can do and what we can measure, if they get worse, we call it loss of function. And so those are sort of catch-all terms for a change that could impact a lot of different things. If we made that part swap and it had absolutely no effect whatsoever, the engine is tolerant to change in that position. It might not run without a distributor, but it doesn't really care if the distributor is kicking out 14 volts or 13.3 volts. As long as it's got something up there, it will run. And so that's an analogy for the amino acid variants we're talking about. So what if we just do a couple of things here? Well, what if we just stick our NMDA machine back in? And rather than swap out a variant, we put the wrong amino acid right there It's in our blue intolerant region. It's right in the region where I showed you those red lines create tension and they sort of pull open the receptor. So that's our analogy. Now we've just put the wrong part right there. And all we need to do to define gain and loss of function in biological terms is just change a couple of words. Now we're not talking about a car. We're just saying, well, we would call a variant, not a part swap, that makes any aspect of the receptor, not the car, work better. That's a gain of function. That receptor now, it's working better. It passes more current when it sees glutamate. And likewise, if the wrong amino acid right there, if that variant made the receptor work less well, we would call that a loss of function. And if the variant had no effect whatsoever, we would say that that's an intolerant position. And so this is one way to sort of think about gain or loss of function. And I think the key concept that confuses people is that word gain and loss. It's really just a catch-all. Anything that makes it work better. If you add a new function, we heard in the first talk, if suddenly the receptors don't just pass current, but they let water follow that current in some way through the hole, and something the normal receptor doesn't do, it picks up a new function. Anything that makes it do more of what it normally does, it becomes gain. Anything that makes it work less effectively becomes loss, including our question, the receptor not being made. It's not going to work as well when it's not there. That's a loss of function. Okay, let's go back to that first electrophysiology slide. So here we have that current again that we just went through. We shock this axon. Here is a terminal. Gold vesicles of glutamate are released. We can see the glutamate cloud here. We can measure it. Tick of glutamate goes up in that cleft, activates these receptors. There's our current. What does gain and loss of function look like in the context of synaptic transmission? Well, here's gain of function. We should have got this much current, but we did that part swap and we got this much current. And so now we have passed far more current than we wanted to. There might have been 100 different ways to make that receptor pass more current, but as long as it's passing more current, the circuit is not going to work right. We could also think about the opposite direction, loss of function. We do the same experiment, shock the terminal, release the glutamate, glutamate rises, activates these receptors, we measure these currents. Instead of getting this much current, all of a sudden we get this much current. And we would call that loss of function. Again, there could be 100 reasons why the receptor is not passing current. The pore could be narrow. It might not want to bind glutamate. The tension on those red linkers I showed you might no longer be sufficient to open it. But they would all have the same result. You'd get less current, and so we call it loss. And then there's our question from just a few minutes ago. You can also have loss if you just don't have as many receptors here. Because, for example, there's a truncation and they're not made. They never reach the synapse. They're never even present to see the glutamate that's released. So got a couple of questions why this is important, and then I'd be happy to take as many questions, just a few more slides. But any questions about that analogy and the general concept of gain or loss?
Just really quick, when you do functional analysis, is that a theoretical test or are you doing it?
No, that's a real world test. We make DNA in a test tube. We either convert it to RNA and inject it into a frog egg, or we trick a mammalian cell growing in a dish to take that DNA up. That receptor covers itself with that cell covers itself with NMDA receptors. We stick an electrode on, we wash glutamate, and we measure in time currents that look very much like that. And so for every variant, and we do 10 assays, 10 cells, 100 recordings, same day, make sure you do the wild type same day, make sure everything's working, times 700 variants. And so it's a lot of work, but there's no other way to do it. And lots of people say, well, can't a computer predict it? Well, a computer can predict it once you know how it works and you program the computer to go off and figure it out. That's a good question. Okay, any other questions about gain or loss? Yeah. have a lot.
Have a lot.
Can have a lot.
One neuron has some that are functional, others that are not.
As a rule, it will. It'll be a probabilistic. If you have one good gene and one gene with a variant, and both of them are making, even if they're not making the same amounts, they're probabilistically assembled, and you'll have some perfectly good receptors and some other receptors that carry the variant, and you'll have a mix. And in sure, and in animal models, when we look at synapses that have that mutation introduced in, we see that mix. We see some with the altered function and some without in the same neuron.
So if you have a nonsense mutation or a truncated mutation, so you can have one neuron that has some
Yes,
properly functioning
yes.
receptors.
Other questions? Say that again. They are, yeah, the turnover rate is, I don't know it offhand, but it's probably on the order of 12 hours or something. Your body is always making these things and recycling them. People tag them with fluorescent dyes, and then in a microscope you can watch movies of the new receptors going up to the synapse and being placed, and the old ones being pulled off and brought in. So it's a very dynamic process. Okay, so why does gain or loss of function matter? Well, eventually there will be therapies for the GRIN variants. It's inevitable. There's a lot of ideas out there now. There's clinical trials. Lots of folks are thinking, how do we fix this genetically? So it's simply a matter of time. And those therapies will come in a lot of flavors. We'll have pharmacology. We'll have medication you take. We'll have some genetic approaches that will eventually be available. There'll be other things we haven't conceptualized. There'll be ways to just simply work with the patients and improve different life skills, and they'll all sort of overlap in this crooked Venn diagram. If we just look at the medication arm, we're going to find drugs that target the NMDA receptor that might be useful in certain patients, and then there'll be other drugs, anticonvulsants that have nothing to do with the NMDA receptor, but some will be found to be a little more effective in GRIN variant patients than others. If we just focus on the drugs that target the NMDA receptor, we're going to have two kinds. We're going to find drugs that increase function, and these are out there, and there are many companies working on these. We're working on these in our lab. And there are going to be drugs that decrease function, and there are many of these out there, some of them clinically approved, others that are currently moving towards clinical trial, particularly Receptor for GRIN 2B. And so now we're in a situation where we have drugs that do opposite things. How are we going to know which variants could potentially be treated with which compounds? Well, it's almost a given that a drug that makes the NMDA receptor work better has a chance at taking a loss of function variation, something that's too small, and bringing it back to where it should be. But this is not what you really would not want to take a drug that blocks the receptor and give it to an individual who already has too little receptor. Likewise, if you were to have an inhibitor of the NMDA receptor, and there are a few cases where these have been shown in the clinic to be useful for a couple of patients. For a gain of function where you have too much current, blocking the NMDA receptor might bring you back, homeostatically to where you want to be, but you certainly would not want to give a drug that makes the receptor work better to someone who already has too much function. And so that gain or loss is sort of a first pass filter as we try to figure out what are we going to do pharmacologically to try to improve the life of the patient. It's sort of a first pass filter on, okay, which drugs are likely to help and not hurt. And then as we go further, the same will apply to genetic solutions. There's all kinds of ways conceptually, not all of them seem practical today, but they may become practical in the near future. There's lots of ways to try to genetically modify cells to either not make them produce less of the receptor, more of the receptor, fix the receptor, replace the receptor. There's a lot of ways to do that, and you're going to need to know what is the variant actually doing before you start thinking about the genetic solution? And so that's why this is actually a really important topic. Now, in the future, in the future, we'll have more complicated functional assessment. We already have it, but it's buried into databases that the different laboratories have, and it's actually too complicated to think about. You kind of have to boil it down and get a gain or a loss. But as we move forward and we have thousands of variants in our databases and many thousands of individuals whose phenotypes we know, clinical phenotypes we know, what we'll probably do is we'll move from gain or loss to beginning to talk about a handful of different properties. I just put these up. These are the sorts of things we measure in our lab. And so down the road, as we come up with even newer approaches to treating these individuals, we may find a way to fix, for example, individuals who just have a problem in the part of the receptor that is real sensitive to magnesium. I haven't talked about that. Or we might find a way to make those receptors that don't go to the surface somehow go to the surface or boost the transport of the good receptors. And so down the road, we'll begin to look at different modalities. But to start with, sort of the starting point is this traditional genetics definition, gain or loss of function. So it is important. Okay, I just wanted to bring up the GRIN portal, and then I'm happy to take any questions. Yes? Yeah, no, that's a great question, and I have like 40 slides behind this talk. We could go into that because I just took a talk and was rearranging, and I shook all those out, and I wouldn't do that to you on a Friday afternoon. You're absolutely right. When we do our analysis, let's say we look at 100 variants, we will find that for GRIN 2B, actually, it's most of the time we get a pretty clear answer. We might find 40 that are crystal clear loss of function. They don't go to the surface. They don't bind glutamate. We activate them and they're there, but we can just get hardly any current out of them. Some of them open and they just open for a blink of an eye and they shut and they don't reopen. We'll find maybe 25 that give you tons of current. Enough current to turn the lights on, and just like so much current you shouldn't see. You know something's wrong, the receptor's open all the time, it's passing a lot of current. And then we'll find the difference. The other 35 will be just what you described. Magnesium block will be down, and so if you look at the receptor, it looks like it's going to pass a huge amount of current, but then you run your assay, did it get to the cell surface? And the frog egg puts it on the cell surface, but the mammalian cell does not, and the neuron does not, and so you realize it doesn't look like it gets there. It might be a loss of function, but if I make it get there, it'll be a gain of function. And so we end up with these mixed ones. And what we've done is we have, to try to get around that, well, I'll just mention it. It might be a little too complicated, but we've actually gone into we've made 100, and we've done it for 2A, not 2B, but we made 100 or more, maybe 140 variants from the healthy population, just from this Nmda database. I think right now this is the only gene it's been done for is GRIN 2A. None of the other rare genes have done this. And we've measured the function of all of those, and we've created a normal distribution. How much can NMDA function change in a healthy person without causing a problem? And what do we find? Well, glutamate potency can vary twofold. Liz is smarter than I am. She's got glutamate binding twofold, but she's just fine. Phil is faster than I am and quicker responses, and his glutamate binds better, but neither of us are up in the clinic. We can do the same thing with all of these other guys. And so what we're trying to do is define the limits in the normal population. Okay, and now when we get a variant, and there's a little bit of a shift, we can say, okay, that's probably not your problem. Yeah, glutamate's less potent, it kind of looks like a loss of function, but that's not statistically different than the healthy population. Or we find out there's a little bit of loss, and you know what? You're just outside the limit of what's normal. That's probably a problem. And when we go back and put that filter on the situation that you mentioned, there's some that are, a few parameters look like they're improved, a few are decremented, a couple of those will drop out of the equation. And so we're in the process of doing that. But invariably, there's a fraction, about a third, that are mixed. And so for those individuals, you're right, gain or loss doesn't help at all. It's unhelpful. But for the others, on the two edges, it is going to be helpful. Yes?
What I'm really here for. So, it seems like this is really really important to know all these particular parameters beyond the loss and the gain function. And first part of the question is there any efforts in the world that you know of that is actively working on developing a model of this right now for the pathogenic, well, actually just for the variants in general?
So, I mean, there is, and actually everybody's working together. The good news for you folks is in the science world, there's always a little bit of friendly competition, right? I work on Nmda Receptor since 1986. I don't know how long that is. It's a long time. And you're always competing with someone who's kind of interested in what you are, and you want to publish before they do, because you know what, if you don't, you're not going to get your grant, and then your chairman is going to tell you we don't have a job for you. So there's always friendly competition. But for the GRIN variants, there's zero competition out there. Every person, I know everybody in the NMDA field, I've been doing it a long time. Everybody is working together. So is anybody doing what you said? Yes, the whole group is doing what you said, and the trick is people are trying to figure out where to put the data, how to harmonize it, how to build a model. And so that sort of leads into this GRIN portal. This is an effort driven by Johannes Lemke, Dennis Lahl, Tim Benke. We're involved. And the goal is not to make a model, but to take all the clinical features of everybody, every single GRIN variant patient, all the clinical features, and then to go back and take everything we've measured and put it on a searchable database, and you can go in here and you can begin to ask questions like, okay, I was talking about magnesium block up here, right? And this is really important. When this goes away, it's always a gain of function unless the receptor doesn't go to the surface. You can go on the portal and you can begin to do research and say, all right, what is that functional phenotype alone connected to? What does that look like clinically? All the kids who have loss of magnesium function, what have they got? All the kids who have loss of glycine potency, what have they got? And so the models will be driven by that. And Dennis Lahl is really a bioinformatics guy who is super interested in this, and he's begun to build models between different ion channels. And Dennis's latest model is he can predict gain and loss of function really well, based on the functional data that we've got. And so there's efforts moving towards that. The first step is to get all the data. And we're in the process of moving, I think about 2/3 of our parameters here. Right now on the GRIN portal, we've got potency, magnesium, we got zinc pH, and that's it. We don't have time course desensitization trafficking. And so right now we're trying to move, I think we have about 500 variants analyzed. We're trying to move all that data back on there. And we don't, and these are harder to measure, so we don't have as much of that. And my prediction would be in 2 years, 75% of the variants that we've worked on, all the data will be on GRIN portal, and maybe 100%. I think at that point, there'll be lots of guys and women who have models of this stuff, and they'll have different models, and they'll be getting up here saying, well, this is what I think works, and the next speaker will say, well, I think that's good, but I like this part. And that's sort of the rich terrain for discovery to take place in. So we're sort of moving in that direction. Sorry, long answer.
Great. And second part of my question is interesting for other parents is how can we get your child to get this kind of analysis done? And
Right. So good question, and there are many groups gearing up to do this. We've worked most with Tim Benke and UC Denver and Johannes Lemke, and they sort of divided the world up. Johannes took a certain number of continents and said, anybody who wants to join a registry from these continents, we'll work you through our privacy concerns, and then the Denver group did the same. I had a young hotshot undergrad write the code to harmonize the two databases. I don't know how to do it, but he wrote REDCap databases. So the German database and the American database are identical, with, as best as you can translate. There's a few words that have ever so slight subtle difference when you're talking about complicated behaviors. And so those two databases are harmonized, and right now we're doing GRIN 1 first, 2B second, and 2A third. We're writing a paper on 185 or 200 GRIN 1 variants, and we're sort of hustling to get all the functional data. Tim and Johannes are getting all the other data. We called a handful of other folks we know and said, we're writing this paper. You're not in the registry, you want to join in? And they all said yes. And so we're sort of working on that. So Tim is going to be here tomorrow, and the way to get in is the gateway for those two databases are Tim and Johannes. And there are other databases. Xavi Altafaj in Barcelona has a database that he has put together. And I know at CHOP, they have a separate database on the GRINs, not nearly as large as Johannes and Tim, but they have a separate database that they keep up. And I'm sure other institutions are as well. Yes?
Just one question. Can you make any comment in terms of like the time in someone's life when you made a difference in impacting phenotypic loss and function? Are there other aspects in brain development that would make a difference if you did it when they're 1 year old or infant stage or adult?
Yeah, it's a great question. It's a question the scientists talk about all the time. There isn't an answer, so anything I say is speculation. I tend to think that there's opportunity for GRIN to be patients early because that gene is expressed very early, and it is higher than 2A up until adolescence. But with opportunity is risk, right? You could make things better by trying to fix it, but it's a delicate window, and we don't really know what's going to happen. For 2A, we're working on a paper right now. It's entirely driven by Johannes Lemke's data. He provided the clinical stuff, and then we have a basic mechanism. But the 2A variants, the null variants, which we had some questions about, those kids have early onset seizures at age 3, but the seizures disappear around adolescence, and they still have issues they're facing. And we went off and did a lot of, looked at a mouse model, did a lot of really complicated neuronal recording, and it turns out those inhibitory interneurons that I mentioned early on, they don't mature in the animals that lack 2A. It's delayed, but it catches up after adolescence. And so those folks have a window of vulnerability. And the idea would be, well, maybe you don't do anything because everything is going to be fine. Well, I would say that they have a window of opportunity to intervene to prevent the effect of having seizures for 10 years on future development. And so there's a window that you could go into. So people are thinking about that, but no clear answers. Other questions? Yeah. So the first thing is I apologize. we are working hard. We're not loafing. So here there's and I talked with Liz a little bit about this at lunch. There's a lot of factors outside of my guys doing the experiment and zipping an email off to Dennis to update the portal. There's legal issues at Emory. When can you report something? When can't you report something? There's grants. There's time. And so we had a backlog. We've done more than 500 variants, including all the good ones. I don't actually know what the total number is. And we've had a backlog of like 40% of those that we have not been able to process. We have partial data sets, and we kind of held them when we developed a new assay saying, well, we don't want to release a premature data set and tell the person it's gain of function, and then we execute the trafficking experiment. We realize, oh my gosh, It doesn't go to the surface. It's not gain of function, it's loss of function, because that's a harder assay. So we held a lot of that data back as we brought online more assays to be complete. And then we also just had logistical backlog. And so there's 200 variants we've done that are not yet been released. We used to give out reports. Emory doesn't want us to give out reports anymore. If you go on the portal, I just checked today, there's a button you can press and it generates a report. And it'll have a clinical report. And so we're trying to clear our data out and move it to the portal and then have automated stuff. And so, so what's the answer to you? Well, the answer to you is the answer to everybody in the room is you should do what parents do. You should persistently advocate for your family members, and you should just pester us, because when we don't answer, it's not out of lack of concern or annoyance, it's because we all get 300 emails a day and they just roll off. So, but you never got the result. Okay. Oh, I see. So we actually did the work for you, but we didn't get an answer. Okay, so that goes back to our question over here. So inconclusive means glutamate potency went up, surface trafficking went down, magnesium changed it a little bit, and we have this equation. We have this equation here where it's not a hard equation, you just run it in Excel. It's basically just algebra where we can calculate what that synaptic current looks like if we knew this changed twofold, this changed 1.5, this went up 2, this went down 2, whatever. We can just plug all those numbers in and out comes a result that pulls them together. And when it's just not clear, it's inconclusive. And so what does that mean? That means it's not gain or loss. But that doesn't mean it's not necessarily pathogenic, because that machine has to work a certain way. And it could be that the timing, it's just not quite working right, but our assays aren't picking up the right feature, and so it could be subtly impacting function. Now, there is one other, if you just bear with me, sorry to be a little bit verbose here, but let me just go all the way back to that picture of a gene. There's one other thing I don't want to fail to mention, and it's really important. We don't know anything about this, and it's 1/3 of the protein. Okay, this is a large. In the protein world, it doesn't fold into anything. It just runs off, all the way over here somewhere, and all kinds of junk hangs on to it. All kinds of signaling proteins hang on to it. That's the part that tells the receptor to go to the synapse, not the soma, not the cell body. And there's all these little sites on here that the body modifies with phosphates and it hangs other, covalent modifications on. And so, but changes in here almost never alter function. Occasionally do. They mainly alter where it goes and how long it stays there. We had a question on turnover. They'll impact how fast the body turns it over. How, will it go to the synapse and stay there, or will it ping in the synapse and then, just go back out of that room and go somewhere else? And so some of these indeterminate ones, the variants, although your variant may not be down here, it might be in a region right here that's kind of impacting what this does. And so there's a whole piece of this that we don't know how to work on. Yes. Yeah. So after the talk, if you know what your variant is, I can put my laptop up when we're done, and let's see. One of the things with the portal is, it's funded, and my IRB, that allows me to take human data and do something with that data, is motivated by the goal of combining functional and clinical data and learning something. And so what that meant when the portal was originally conceived of and we started building it is, if the clinical data exists but the functional data exists, it didn't go on. And if the functional data existed but the clinical report was incomplete, it didn't go on the portal. And so it could be you're not finding it because we have a partial data set. And we've been actually meeting the last couple months. We're just trying to do a data dump and get everything we've got on there now. And so there could be some issues in how we originally started to populate those databases. But don't let me leave here without opening it up, and we'll have a little search and see if we can find it. Other questions? Yeah. That's it right now, but that's better than it was a few years ago. There's L-serine and D-serine for loss, and there are multiple pharmaceutical companies advancing NMDA receptor potentiators, but and they've been doing this for 10 years, but they want to be really cautious because that potentiator is like a gain-of-function variant. And a lot of them in the animals will produce seizures. And so everyone's super careful about, before a potentiator goes to the clinic and a couple of the patients end up with seizures, let's figure out what we're doing. So those are all moving forward. Multiple large, multinational, huge companies have programs, and there's compounds out there, and they are moving forward. For the blockers, Receptor is the 2B inhibitor. It's a prototype of a class that all companies had worked on, and so there's a lot of 2B work going on right now. Receptor will be the first one that would be available for pediatrics. And there's memantine. There's also some patients have been on subanesthetic ketamine. Some of the 2D kids that worked really well, but those kids had a lot of challenges. And then there's a Harvard patient who dextromethorphan worked really well. There's a paper that came out, in not necessarily one that parents would run off and be dying to read at night, but it was the structure of the memantine binding site. And we're working on a predictive algorithm that'll tell us which variants will make memantine more potent. And when memantine's more potent, it actually will block the variant receptor, not the normal receptor, and that should be a way of figuring out why it works in some kids and not others. And so that might have a little bit of some clarity about when to use it. There are other NMDA inhibitors also being developed in large pharma that are, they're all sort of marching forward slowly. The problem with inhibitors is they cause psychosis like PCP, and so just like the seizures with the potentiators, companies are super careful about when they're going to advance a inhibitor. But there's a lot of NMDA work going on right now. I mean, I interact with a lot of these companies. Everybody's got a program. So I think there's hope that things will come out there that are new, and not just in your lifetime, in the next decade. Yeah. Yeah, I didn't hear it. You want to? Yeah, just soft voices.
So, I recently read the paper about rescuing green 1 mice, and then is it likely explaining that green 2B will also have the similar result, or is that green 1 and green 2B totally different one?
Yeah, that's a great question. So that's referring to a paper from Amy Ramsey, where she has a mouse made decades ago that for some reason just expresses maybe 20% of the GRIN1 that it should. And everybody I know assumed that mouse couldn't be fixed, has a lot of deficits, because we assumed once the circuits are not wired correctly, you can't unwire them and put them back together. And it turns out Amy showed that supplementing the animal late brings back function. So that was a really exciting result. It was an unexpected result. It's a very encouraging result. It sort of emphasizes how plastic the brain is. You know, the brain has all these mechanisms try to fix itself. You know, if you watch a stroke patient retrain, they end up gaining function you would never thought they might. And so I think it's hopeful. I think it might encourage more folks to actually try that experiment and see if they can rectify problems with reduced 2B function. There is a problem with the 2B mice. The mice that lack 2B are very difficult to raise. And so there's like a logistical problem and doesn't, you know, speak at all to humans. It's just a mouse thing. So, but I hope it is. It kind of changed my mind, because I would have predicted no way you can fix these guys as an adult that lacked the receptor their whole life. And so I'm encouraged.
Yep. Thank you to Dr. Traynelis for his presentation. Give him a round of applause, please. Thank you so much.